Good morning, everyone, and welcome to this session. It's my pleasure to introduce the keynote speaker, Alejandra Capozzo. Has a degree in biological sciences, specialized in biotechnology, and a PhD in biological sciences from the University of Buenos Aires. She holds a postdoctoral degree in neonatal immunology from the University of Maryland. She has a specialization in immunology from the National Academy of Medicine of Buenos Aires, focused on vaccinology and diagnostics. Previously, she worked as market development manager for Latin America at Prionics and as head of research and development at Biogenesis Bagel. She currently works as a principal researcher at CONICET since 2018 and is in charge of the Applied Veterinary Immunology Laboratory at the Institute of Virology of INTER in Buenos Aires. She coordinates research on viral diseases that affect reproduction within INTER's National Animal Health Project and is also a member of the Directive Committee of the Argentine Association of Veterinary Immunology. In addition, she is the researcher of numerous national and international projects with collaborative ties with Europe, Africa, and Australia. She has been working on foot and mouth disease for 25 years. Alejandra will answer questions at the end of the diagnostic sessions, but please type your questions in the chat box and then she'll pick them up later. Thank you.
Good morning. Today we're going to talk about the serological assessment of anti-Fmdv antibodies. We're going to go through the quantity, quality, and all the differences we have found between species along the years. And the first question you ask is why measuring anti-Fmdv antibodies? For vaccine release to the market without the need of infecting animals, and that's really important. For vaccination surveillance and herd immunity to assess vaccine efficacy in the field, to predict homologous or cross protection, so to predict protection against the strain that is included in the vaccine or against a strain that is not included in the vaccine, and also for vaccine efficacy trials when you want to try current or novel vaccines as well. And for most of these actions, you need validation with a challenge test. And this is also very important because all the time you need to challenge the animal to see if these measurements you are doing are actually working. So serology is a very important part of the vaccine control. In every step of when you have the final product, then you need to know which strain to use, how will that work, and then you have to control what's going on in the field. And for all that, you need serology. So serology is quite important. And the information that serology provides us right now with the currently used assays are just two. One is the virus neutralization test that is a gold standard that quantifies antibodies that impede virus infection of culture cells. And the other ones we use is the liquid phase blocking ELISA or the solid phase blocking ELISA. And this blocking ELISA estimates total antibody levels. So we are just measuring, quantifying neutralizing antibodies or total antibodies. And we have been doing that for 35, 40 years, something like that. If you go to the history and you go to PubMed, you will find that the liquid phase blocking ELISA was developed in the late 80s. There's those papers that I mentioned there. They used, in most cases, purified live virus or live virus PEG precipitated. And as you can see here, this correlation between the VNTs and the liquid phase blocking ELISA was demonstrated quite long ago using, this is from this paper, this live virus. So, and of course, they suggested, these authors so long ago, that if you have this kind of assay, is much more beneficial than using the VNT because, of course, it's more and more reproducible and you may still predict protection. So many people have been using the liquid phase blocking ELISA along the years, and Argentina is a very good example of that. So they have performed validation of this liquid phase blocking ELISA against expected protection. So along the years, many challenge trials have been taken had been done, and in each trial, samples were taken 60 days post-vaccination, and then animals were challenged 90 days post-vaccination. And then you see who were protected, who were not. And then you can build these very nice correlation curves, and you get even a cutoff number of what my colleagues called the expected protection, 75%. So that's the antibody titer that would protect 70% of the population. If you vaccinate, you bleed at 60 days post-vaccination, and you challenge at 90. So if you have these vaccine strains and you work just with those, this works really well. So let's go to see how VNT and liquid phase blocking ELISA work with homologous protection in cattle. And this is a nice experiment where animals received an A24 vaccine. These are several experiments with the same vaccine that was aging. So you have some animals that were not protected, even this is a very good vaccine. And then they were segregated in their regarding their VNTs or the liquid phase blocking ELISA titers. And as you can see here, you get a very broad range of values in the protected animals, even by the liquid phase or the VNT. And if you see here in the VNT, you have several animals that have VNT titers that are almost exactly the same than the ones that were non-protected. And if you go to this part of the graph, you will see that you have a lot of animals with high total antibody levels that are exactly the same that you find in the protected animals. So when you put the 75% expected protection cutoff for each of the assays, you see that you are not really predicting very well. You have all these animals with a very low VNT titers that are protected. And here you have a lot of dispersion. So how accurately are we predicting protection using only this assay? And then we did just the statistics to see what's going on, how is the concordance between the VNT and the liquid phase blocking ELISA with protection, what's the sensitivity and specificity, and this is what we found. We found that the concordance with the PPG, that is the challenge test, is really not very good. It's between, well, the kappa value will be between good and fair, so it's not really high. And then when you go to the specificity and the sensitivity of both assays, you see that the values are not very impressive, as you could see for the last graph I show you. But the thing is that you have like 80% specificity for the VNT and 80% sensitivity for the liquid phase blocking ELISA. They are like seeing different things. But the accuracy to predict that an animal will be protected is about 80% for both of them. So the concordance that these assays have with protection is really low. So really, we are not having a very good assay to measure protection if we just stick to one of them. They are measuring different things. So the thing is that what we have is the virus neutralization test that everyone knows is cumbersome, uses live virus, it's not high throughput, no one wants it. And we have this liquid phase blocking ELISA to replace it, to measure total antibodies. But there is an issue with this ELISA. Although they are using it, for example, here in Argentina with the vaccine strains. You need to prepare 2 antibodies every time and for each strain. So for a vaccine strain, that's fine because that's the only one you have. But if you have to prepare it for each field strain, it takes quite a lot of time. You have to immunize animals, prepare the serum, standardize it. That's an issue. The other issue is that you capture the antigen with these capture antibodies. So you may be also capturing disrupted antibodies. And we all know that antibodies that are directed to whatever epitopes are inside the capsid are not protected. So do we need alternative ELISAs? I think we do. We all agree we need an ELISA, and I won't go through all this list, but low middle income countries use ELISA, and that's why we focus on ELISA, because it's an assay that you can do in the lab, that you got numbers out of it, and low middle income countries are using it. So we went through ELISAs. But then we decided, well, what type of ELISA we're going to develop, and what is it that we're going to do with it? So the first thing that we pay attention to is the capsid structure, because we know, and it has been published before, that this whole viral particle is protective, but when it's disrupted, when you have the 12S particles, then it's not protective. So the thing is that what antigen you're sticking on the plate, right? You need to bind on the plate something that would measure antibodies directly to whatever is a protective antigen. So in a vaccine, you usually have these whole particles, but sometimes, and usually they start to get disrupted because of aging or because of the strains. Some strains are not that stable. So you get also these 12S particles. So these 12S particles will be exposing internal epitopes of the viral particles. So in the sample, you will have both. You will have antibodies against internal epitopes that are not protective, and those against exposed epitopes that are more likely to be protective. And if you do serology, you have to be very careful what you measure, because if you are using these particles, you will get some of these antibodies to exposed epitopes, but you are also measuring those that are binding to whatever epitope is inside the virus. So that will not be that related to the neutralization. And in fact, the neutralization assay will measure antibodies that block viral entry to the cells, viral infection to the cells, but are not detecting these antibodies directly to internal epitopes. The liquid phase blocking ELISA, as you put, just what we do now is we take virus from the supernatant of infected cells, we inactivate it, and we use that. And there you have everything. You have 12S and you have the whole particles. So you measure antibodies against everything, and then you capture them in the solid phase. So you measure antibodies against exposed and internal epitopes as well. So we thought, why don't we go with an indirect ELISA, and we use purified virus, and then we are not measuring those antibodies we don't want to measure. And this is not a very original idea. In fact, well, this demonstration that when you have these 12S particles, you get these mixed antibodies that do not relate to neutralization was demonstrated in 1981. And you want to measure these exposed epitopes, and this indirect ELISA using purified viruses bound to the plate, that was developed also even before, when I was a very, very, very little girl in 1978. So we knew that we have to do something with that, but we need to be sure that we were on the road. So what we first did was to immunize pigs, in this case, with an aged vaccine, an old vaccine that has 45% of the vaccine, of the antigen from O campus, was already disrupted because it was an aged vaccine. And the A24 was complete in 90%. And this is because of the inherent stability of these viral strains. So for the A24 antigen, we got a lot of whole particles. For the O1 antigen, we have whole particles and disrupted particles as well. It's like 50-50 of each. And we wanted to see if we can differentiate this kind of antibodies by serology. So we vaccinated the pigs, and we took blood samples, and we ran the liquid phase blocking ELISA, indirect ELISA, and also, of course, the VNTs. And what we found is that, as you can see here, if you measure total antibodies, the assay does not discriminate between those antibodies that are directed to exposed or non-exposed epitopes, or to the 12S or to the 140S. For them, there are antibodies, measure total antibodies. But when you do the indirect ELISA or the VNT, when you are sticking on the plate the whole particle in the case of the indirect ELISA, or when you are blocking infection in the case of the virus neutralization test, then you can differentiate if your vaccine was disrupted or not, or if your antibodies are more related to protection or not. So when we did all this correlation, as the one I showed you before, that was done long ago, and then we did it again with these samples, we see that the correlation for O campus, that is the disrupted antigen, is not that good, and it's definitely much better for A24 Cruzeiro, where the animals received whole viral particles. So The liquid phase blocking ELISA is using non-purified antigen. You just take the antigen from the supernatant of infected cells, and there you have everything. So what we did was to purify this antigen to see if you are using purified antigen and you ensure that you have whole viral particles, then will the correlation be better as it has been before? And the answer is yes. When you purify the antigen, the correlation moves from 0.47 to 0.72. Look at this. And here it gets better as well. So you are really measuring antibodies against exposed epitopes here. And that's the difference. When you are not using a purified antigen in your liquid phase blocking ELISA, you are measuring the blue and red antibodies. When you purify, you just measure antibodies to exposed epitopes, and that's what you want to measure. And the thing is that when we do the liquid phase blocking ELISA, we used to inactivate it now. It was not like in the 80s where they used the live virus. We inactivate the virus. And sometimes the inactivation process itself with the VEI takes like 24 hours. And sometimes it stays long at 37 degrees. And look here what we published some years ago. We showed that if you put, for example, O1 campus 24 hours at 37 degrees, you lose almost 80% of your whole viral particles. So we have to be really careful on what's the antigen we are using in the liquid phase blocking ELISA. And you get better correlations when the whole particles are used. So its particle stability is really important to measure antibodies against exposed epitope. And of course, you avoid the use of detector of capture antibodies if you have an indirect ELISA. Of course, you will tell me, okay, you said it was already developed in the late 70s. And I say, oh, yes, okay, 43 years ago, that's true. But the bottleneck and why everyone changed was purifying the viral particles. So we took that as well, because of course, when you have to purify viral particles, you have a very long and cumbersome and expensive way to go with the ultracentrifugation, and you have to do the sucrose gradient, and you have to harvest the gradient, and you have to pellet the virus, and then you get the counted antigen. So luckily, some years ago, a friend of mine who was working at Biogenesis, even when I was working there as well, developed the size exclusion chromatography to purify the whole viral particles. And this is just, you concentrate the antigen using PEG, and you go through the column and you get the antigen ready to use. And when we tried that, the purification method by size exclusion chromatography, what we found is that compared to the particles purified by sucrose gradient, they work exactly the same. You can see here in pink and in blue, they are different samples that were used as controls, and you can see they behave exactly the same. You get the same OLG values. So this size exclusion chromatography produces a ready-to-use antigen, whole viral particle antigen that can be used as a coating agent, and you can do it without any further processing. So format solved, we have indirect ELISA using whole purified viral particles. But the idea is, which are the immune parameters we need to evaluate? So we are now measuring neutralizing antibodies. We also measure total antibodies, but you have a whole range of factors in the immune response. So what are we going to measure? And to answer that question, we also needed to find something we can measure using a very simple ELISA. Otherwise, it won't be useful. So what we know is that when the immune response matures over time, we have, after the antigen recognition, the lymphocyte activation and proliferation, and then you have a phase of somatic hypermutation, where you get antibody, of course, antibody secretion and memory B cells. But this is where we focus. You also get isotype switching, so you move from IgM to IgG1 or IgG2, okay? And also the affinity of the antibody matures. That means that the antibody you have at the beginning is less strongly bound to the antigen than the antibody you get as the immune response matures. So if you see these curves, you will see in all the books with the primary and the secondary immune response after infection. What we are doing is to see what's going on about 21, 30 days after vaccination. considering that we need to have a mature immune response. So when infection comes, you will have this very nice protective secondary response. So the antibody avidity, and I won't go very deep on this because I've been talking about avidity for years and years, is the sum of the affinities of the antibodies. So the affinities define epitope, paratope binding strength. So it's just 1-to-1. But the avidity is a property of the serum because the antigen is bound to several parts of the antibody, and then you get a network that is the binding strength of that whole serum sample, and that is called the avidity of the sample. And the avidity of the sample is very easy to measure, good for us, using ELISA. So the parameters that we decided to measure were isotypes, IgG1, IgG2, and IgG avidity as well. And how is the design? The design is very simple. Of course, we're using an indirect ELISA that works, binding the whole viral particles to the plate. And we know that this indirect ELISA gets rid of these non-protective antibodies. And then we combine with a conjugate that can detect, for example, IgG1 or IgG2 or IgM. You can measure whatever you want. And what about the avidity ELISA? Well, the avidity ELISA is the same format. It's an ELISA plate where you put your serum sample in duplicate wells. Instead of using just one, you use 2 of them. And you can see here, I just tried to paint in pink those antibodies that are low binders. They are not bound to the antigen very strongly. The binding is with non-covalent forces. So if you wash with urea, if you wash with PBS, you don't detach these low binding antibodies. But if you use a chiotropic agent that can be, for example, urea, then you detach that low avidity binder. You do it, and then when you do that, you can come then with a conjugate, you get a color, and you can relate those values, and you get what we call an avidity index. The avidity index will go up when the immune maturation occurs. So if the immune response is more mature, you will get more avidity. And that's the basic idea of the assay. That's absolutely simple. So let's start analyzing the homologous immune response, and let's go through the isotype responses. I'm not going to show you all the results because this was started long ago in 1997. I was still a student, and this was my very first paper, where I demonstrated that animals with low total antibody titers, those measured by liquid phase blocking ELISA, but high levels of IgG1 were protected against infection. And that was done with a huge amount of animals. That's a very nice paper that I published very long ago. And then some years ago, not that far, another student in Mariano's lab and showed that the IgG1 is the main isotype that is induced after infection as well. So if you see the isotype responses after vaccination, you will see that in cattle, you have the IgM that never seems to go down very quickly, but you have a peak of IgG1 right away, even at 7 days post-vaccination, you already have it, followed by IgG2 that goes more slowly afterwards. In swine, the things are different. You have the peak of IgM as expected, and then you have IgG1 and 2 that has more or less the same kinetics. So between species, the kinetics of isotype responses are different. Okay, so we have the isotype ELISAs, we have the avidity ELISAs, and we wanted to know if they were providing or not different or additional information to the VNT. And a nice way to do it is to make the correlations and study the concordance between virus neutralizing titers and the readout of the assay. What I have put here is we have the different assays. This is the same vaccination trial I showed you before with protected and not protected animals. And we have the avidity ELISA, the liquid phase blocking ELISA, and the isotype ELISA. And here shaded in gray is the cutoff value for the VNT for this particular H24 Crusader strain. And I just aligned the different assays regarding their concordance with the neutralizing antibody titers. And you can see that antibody avidity is quite high. The concordance is quite high with the VNT, so it's more or less providing the same information. And then the concordance goes down, and clearly these isotypes are giving a different kind of information. You can see that even animals with titers in between 1.5 and 2, they already have high levels of IgG1, and the IgG2 levels are lower, as we have seen before. So more work is needed, but we can exercise the potential benefit of replacing, for example, the VNT with the avidity, and then complement information with the IgG1 and 2 ELISAs. We don't need to use the total antibodies perhaps anymore. We also studied the correlation between VNT with liquid phase blocking ELISA and avidity in buffaloes. And we are having a poster showing this and even the difference with maternal immunity. But what I can tell you is that while avidity has a very nice correlation with the VNT, the liquid phase blocking ELISA couldn't discriminate between undetectable and even high VNT titers. So it doesn't seem to be working very well for buffaloes, maternal immunity. Let's move then to heterologous protection. How does avidity and isotypes responses predict cross-protection, or what we call heterologous protection? So what is cross-protection? Well, the question we want to answer is a specific vaccine likely to protect against a specific field strain? That's the question we're asking when we study cross-protection. And what we do right now is to use a vaccine containing one antigen, call it A. You vaccinate the animals, and then you take samples for serology and you challenge, and you do serology and challenge against this other strain, call it B. And then you see if from serology you can predict protection that you can see in challenge. And the way we do it now is using what is called the R1 value, that is a ratio between the titers against the field and the vaccine strain. And the cutoff has been fixed in 0.3. And the thing is, when you use these values, can you really predict protection? And the answer is no. And we did an heterologous challenge study some time ago, and this was performed quite long ago, and it has been published. So I want to go through it just very quickly. We got 47 animals with symptoms and 17 that were protected when animals vaccinated with an A24 vaccine were challenged with A Argentina 2001, that is an heterologous strain. And the thing is, when we plot the VNTs against the vaccine and the field strain, we find protected animals that have very low R value, and they will be predicted as non-protected. And we have also the other way around. So the R1 values were not working. But if you see, for example, the avidity and the IgG1-2 titers, this is avidity or IgG1-2 ratio, we can really differentiate protected and non-protected animals. So that seems to be the way. We then analyze, and this is all published, and I have talked about this like several times. If you put all the assays together, all of them, the R1s, the isotypes, the VNTs, and everything, we saw the specificity of all these assays in heterologous protection was good, but the problem was in sensitivity. So the sensitivity was awful. You have a 0.12 sensitivity for some R1 values, for example. And the IgG1-2 ratio. So we found that combining different assays actually improved the accuracy of the serology because sensitivity is low. So we exercise conditional testing. Conditional testing is to evaluate a serum sample using a screening test and then assess those samples that are not detected as protected in a second test. Samples that are determined as protected by either of the tests were classified as protected. And doing that, we found that we have very high accuracy levels for detecting cross-protected animals that were over 90%. It worked really well. And in fact, I also invite you to the oral session 7 on Wednesday by Mariano, where he's going to show you that if you have different vaccine formulations against A24 strain, and you challenge with A2001, you really cannot differentiate protected from not protected by using total antibodies, neutralizing antibodies. You can also only measure protection by using avidity. So to conclude, antibody responses to currently used vaccines differ between species. The use of purified whole viral particles improves the assessment of antibodies against exposed epitope. The liquid phase blocking ELISA can be an alternative for vaccine control. Of course, you need to be really sure that you are using whole particles. However, it's not very convenient for arising field strains. Liquid phase blocking ELISA is not suitable for buffaloes. I invite you to the poster. You will see more details about that. And avidity seems to be an acceptable alternative to the BNT with the benefits of being a simple single dilution indirect ELISA. And what we saw is that protection, even homologous or heterologous, can be inferred by analyzing at least two aspects of the immune response. For example, avidity and isotype titers. So our message is that several aspects of the antibody response should be considered to provide an accurate estimation of protection. So now we have not only the currently used, but we have these new assays using purified inactivated viral particles that can give us more information, and we need to correlate them with the challenge studies. So we now have simple tools to possibly predict homologous and heterologous protective responses. And would you like to try them in your challenge experiments? If you would like to do that, you can write to me. Thank you very much.